I'm really thrilled to be speaking to Krista tonight, and really appreciate you taking time, Krista, from your busy schedule. I know imaging has kept you out of trouble these days, right? So first of all, congratulations on your renewal by the NIH for imaging. It's such an important study. So I'd love for you to just lay the groundwork of imaging. I often think of imaging as the 3 sites in the U.S. Tell me about those 3 sites and how you've thought about how you have expanded your interest way beyond the United States.
Hi, Pat. First of all, thank you very much for spending some time with me today. And I'm just delighted to be part of the PPMD conference again. I really appreciate it. So imaging DMD has grown a lot over the last 10 years. As you know, we started out with 3 imaging sites. There was our partners in Portland, Oregon Health Science University, Children's Hospital of Philadelphia, and then University of Florida. But we've really expanded very broadly. We've really tried very hard to move a lot of these measurements across many different sites. And at this point, we have about 30 imaging sites across the United States and Europe that are collecting data in the same way as we have in our original study. So a lot of expansion, and really with the goal to be able to run and introduce MR imaging into clinical trials.
Yeah, thank you, Krista. I know so many families over those 10 years that have participated twice a year, and really, first of all, look forward to your wonderful staff and participating. A couple things. I know at first when you started out, you were imaging the quadriceps muscle and basically the lower leg, and now you're looking at many more muscles, the trunk, the upper arms. Are you seeing differences, significant differences in how these muscles are changing over time?
That's a very good question, Pat. We've learned a lot, as you know, by having the opportunity to work with so many families. That really helped us understand the disease and how the disease affects different muscles. So whereas we collected about 10-year natural history data in the lower extremity muscles, we've expanded to the upper extremity muscles. We're also measuring the respiratory muscles, particularly as the boys get older. We know that it impacts their pulmonary function. And so we really need better measurements there as well. What we've learned is that this pattern of fat infiltration happens across the different muscles, but the rate at which it happens is different. The reason that's really important is that, as you know, we're trying to use MR as potential biomarkers in clinical trials. So if we're looking at younger age groups, then maybe the quadriceps is a better muscle to target. But if we're looking at older boys that are non-ambulant, then there's other muscles such as respiratory muscles or the upper extremity muscles that may be much more valuable. But at the end of the day, what's really important is that this measurement of fat infiltration seems to be a really good measure of disease progression and a valuable endpoint that we can potentially use in clinical trials.
So I think that you're on the process to qualify fat fraction as a biomarker. So are you qualifying, so where are you in the process? And second question is, in terms of, are you qualifying that in a particular muscle or, across the board in terms of many muscles?
Yeah. So what we're doing right now is we're really building on all of the natural history data that we have collected. Our richest data set are in the lower extremity muscles. So for our efforts to move forward in MR biomarker development and qualification, it's really important that you have a rich data set. So our focus has been on fat fraction in 2 lower extremity muscles. That's the vastus lateralis, which is one of the quadriceps muscles, and then the soleus, which is one of the lower leg muscles. So we put together a steering committee, which is an MR imaging steering committee, which has representatives from PPMD, from industry, from NIH, and from academia, really working together to understand the data, and then collaboration with CPAD, that is our partner to look at the regulatory component. We're submitting an application, a letter of intent right now to the FDA. So a lot of work has gone into it, and our primary focus right now is the lower extremity muscles, just because that's the richest data set that we have.
I see. So Critical Path has the D-Risk Consortium. So this is a consortia that has a number of data sets within it, looking at disease progression models and trial simulation tools. So can you tell me a little bit about the path to qualify this? Is it, I'm sure it's very complicated because it's an FDA qualification, and so that takes a period of time. But at the end of the day, when we're looking at gene therapy as an example, one of the questions is, how are you going to get that approved, and what does it mean in muscle, and how will we know? Because obviously it was based on people who were doing very well from birth to their very older ages. So this microdystrophin will be hard to figure out unless we have such a validated tool to say the fat fraction has not changed or has changed, or the rate of change. So can you give us a timeline of that, Krista, and what you see into the future?
Yeah. You're absolutely right, Pat. It's a complex process. It's been a lengthy process. So I'm certainly by no means the expert in regulatory approval, and our team is learning as we go as well. But certainly our partnership with Critical Path and D-Risk has been really, really important. They have a lot of experience in this area. So our first step is a submission of a letter of intent, which has been pretty much completed and is ready for submission to the FDA. At that point, we hope to hear back from them. So what we're actually submitting is a letter of intent for a fit-for-purpose application that allows us to really submit data and submit a briefing document that describes the disease trajectory of fat fraction and its relationship to function. What has been really important in the work is not just that MR is sensitive and it tracks disease progression, but ultimately that it is closely related or is associated with functional endpoints, and it can predict future function. So in collaboration with Bill Rooney and Sarah Kim and others, we have really invested a lot of effort to understanding how well MR biomarkers predict future changes in function. And that's ultimately really important because the MR measurements will tell us, hopefully quicker, that a drug may be working or not, but you ultimately want to know that that has functional implications. And that's really what the goal is of this project.
Great. So, and Krista, the study is still open for people to join this study, is that right?
So right now, the study was renewed over the last year. We have changed the direction a little bit in that the sense that we will continue to develop these measurements. We're focusing now more on respiratory muscles in non-ambulant patients. As you know, that is a group that particularly is in need of more outcome measures. And follow up to some of the discussions earlier, we're also looking at Becker's patients. There is a real need to understand the natural history of Becker's as well as having more measurements. So that will be the focus. So we've expanded. We are looking at both Duchenne as well as Becker's patients.
And you're also including female manifesting carriers?
Yes. We are doing a separate small study that is going to focus, thanks to your support, Pat. We really appreciate that. We are going to be looking at female carriers. We've done a little bit work, but we're going to expand on that as well.
Good. Krista, I'm really so thrilled about imaging DMD and what it can bring to this community, a highly sensitive biomarker that really can predict function, that isn't biased, right? That isn't based on someone's opinion. And I think that your work in both ambulatory and non-ambulatory and Becker, and certainly manifesting carriers, is remarkable. So we're thrilled to be a part of imaging, and for anyone listening, joining an imaging study I think is so critical to progress. I can't thank you enough for spending time with us tonight. We so appreciate all of you, and your team is such a great team. It's a pleasure to work with you. And so next year we'll be together in Scottsdale. So thank you very much for joining us tonight, Krista.
Thank you, and thank you to all of the families.
Thank you.